I wanted to take a minute and talk about how the eye develops. So as you see here, this is a fish embryo, and this is the developing neural tube right here. So as we move this forward, okay, you can see the neural tube start to mature. You see over here the optic vesicles start to bud out. This is from the diencephalon. Okay, so let's move this along a little bit further. You see those? So the eye cups are starting to form out of the diencephalon, which is true in vertebrate embryology. And so let's take a look here at the chicken. Same type of thing. You have your neural tube. Here you have your optic vesicles that have budded off from the diencephalon here. You see how they push outward to develop the eye cup there. And here you have it in a form a little bit more familiar to you. So if you remember from the embryology video, that this is your neural plate here that then starts to fold up upon itself and become the neural groove, which eventually then becomes the neural tube. And so you see here as it moves up on itself, right here is where the optic grooves start to form. So as it starts to close here, as the neural tube starts to close, your optic grooves start to push out in either direction from the diencephalon and then move up. Here you see it forms the optic cup here, and this portion forms the retina. So what we have here in reality is an eye that really is part of the central nervous system. It's part of the diencephalon that has kind of pushed out and forward. And so what you see here, the optic nerve, really in reality is not a nerve in the true sense of a nerve. Rather, it really is truly a white matter tract in the central nervous system, although we refer to it as a nerve. Given that this is still part of the diencephalon, the axon fibers coming off of the retina then would become a white matter tract in the central nervous system. Let's take a look at the eye. So here's the eye here. This is the back of the eye, the front of the eye with the lens. This is your retina back here, the optic nerve coming through here. So light comes in, hits the lens of the eye, and is refracted back to the retina. Light then has to travel through several layers of cell to actually get to the phototransduction cells that then will provide or produce a signal. So down here you have what's called the retinal pigment epithelium. This is a single cell layer. Cells are densely packed with pigment granules, the point of which is to protect the eye or shield the eye from excess light coming in. The retinal pigment epithelium supplies omega-3 fatty acids for building membranes. It supplies glucose and amino acids. Problems with the retinal pigment epithelium have give rise to disorders such as age-related macular degeneration and retinitis pigmentosa. So this layer here, comprised of rods and cones, is where the light is headed. This is what gives rise to vision. Before the light is able to get to this layer of cells, this layer of rods and cones, must go through a number of layers of cells, the first being the retinal ganglion cells, which we'll talk about at great length later. Then you have a layer of bipolar cells. You have Amerkin cells, seen in green here, and horizontal cells. Now here we're looking at this layer here, which is pretty much consistent with most of the retina, with the exception of this small area here called the fovea. As I mentioned earlier, rods and cones are the phototransducing cells of the retina. So rods transduce light, and rods are found pretty much dispersed widely across the retina. Cones are responsible for color vision and also for visual acuity, for being able to discriminate fine details in vision. And although cones are somewhat dispersed out here, they're heavily concentrated in the fovea, and primarily for visual acuity and for color vision. Such that what you see in the fovea here is predominantly, or exclusively actually, cone cells, whereas when you move out into the periphery, you get much more rods and just a few cones interspersed. And in the retina, there's about 120 million rod cells and about 6 to 7 million cones. And you have three types of cone cells, and I'm not going to get into them at great length right now, but you have red cone cells, which comprise about 65%, let's say 65% of the retinal cone cells. You have green, which comprise about 30, somewhere between 30 and 32%. And then you have blue cones, which are the rest. So a small proportion are the blue cones. Interestingly, the red and green cones are found predominantly in the fovea, remember that region here, the blue cones actually are scattered around the outer areas of the retina. And when we talk about color cones, when we talk about red cones and green cones and blue cones, I hope you understand that they're not really red, green, and blue, that they are actually able to absorb wavelengths that are in the red spectrum, in the green spectrum, and in the blue spectrum. Now, because of the cytoarchitecture of the retina with the ganglion cells, let me go back to my pointer there. So the ganglion cells are located again out in the, actually more towards the surface of the eye, and then you go down deeper to the photoreceptor cells. So signals transduced to the ganglion cells that then send axons into the optic nerve and back into the brain. And so you have this area here at the optic disc that actually contains no photoreceptors whatsoever. And so what's interesting is that this actually is a blind spot, such that if you look at the you cover one eye, so let's say you cover your right eye, and you look at the plus sign with your left eye, and you kind of move toward it and move away from it, at some point this black circle will disappear. And that's due to your blind spot. And what happens is, so the vision information goes back to your brain, and your brain kind of fills in that blind spot for you. Now let's take a look at phototransduction with rod cells. So it's a bit counterintuitive. So in the dark, actually, the rod cells are depolarized. So there are currents of potassium and sodium both in the rod cells. There's potassium going out, there's sodium coming in. Essentially, in the dark, you have a depolarization of about minus 40. So that in the dark, this rod cell is depolarized, and you have the neurotransmitter glutamate being released at the synaptic cleft on this bipolar cell. So interestingly, although glutamate is traditionally considered an excitatory neurotransmitter, on bipolar cells, it's actually inhibitory, so that these bipolar cells are off. And what you have is then decreased transmitter release on the ganglion cell, which is sending projections back into the brain. When the light comes on, these rod cells become hyperpolarized, meaning they get shut off, so there's no transmitter being released. And all of a sudden then, this bipolar cell turns on, releases transmitter, that then turns on this ganglion cell that then sends signals to the brain. Now, these amacrine cells that we talked about earlier are at the inner plexiform layer, and they actually communicate a great deal with these retinal ganglion cells. In fact, about 70% of the communication with the retinal ganglion cells come from the amacrine cells, and the rest come from the bipolar cells. So they play kind of an intermediary effect, fine-tuning the signals that are coming in. Also, with the bipolar cells, you see a lot of connections across, particularly the rods. So you see one bipolar cell may connect with a number of different rods. There appears to be a greater one-to-one connection of bipolar cells with cone cells, and especially in the fovea, than outside of the fovea. So you have, let's see, about 260 photoreceptors. And then you narrow down to about, this is both eyes, about 2 million, I'm sorry, it's 260 million. And then you narrow down to about 2 million ganglion cells. So you have a lot of information coming into these cells and a lot of information coming out of 260, 130 per eye photoreceptor cells that then is kind of being modulated by the bipolar cells and by the amacrine cells, and narrows down further to about 1.5 million per eye, or 2 to 3 million ganglion cells. So you see a lot of condensation of information that then needs to go to the brain and branch back out to recapitulate what has happened in the visual field. From your book, we see an illustration of the brain. So this is the occipital lobe, where you find the primary visual cortex. Remember, this is towards the front of the brain. Here you have your eyes here. You have the visual field here. So you're looking down, straight down at half of the brain. Here you have your left visual field and your right visual field. And because of the curving, this is so interesting with the eyes, because of the curving of the cornea here, the retinal layer here, you have light going in from the right visual field to both eyes. You have right visual field going over to the temporal region of your left retina and to the nasal region of your right retina. So again, right visual field goes to the left side of your left eye and the left side of your right eye. Left visual field goes over to your temporal right area of the right eye and to the nasal portion of the left eye, so that you have all of the information from your right visual field coming down here. From this eye, it crosses at a feature we call the optic chiasm, and it's very distinct when you look at brain tissue. So the right visual field information hits the nasal retina here. That information is carried over to the contralateral, meaning the other side, the contralateral side, where it joins with right visual field information from the left eye to go back to, and this is important that you know this, the lateral geniculate nucleus of the thalamus. And likewise, in your left visual field, then, the same thing happens. You have your left visual field going over to the nasal portion of the left retina and over to the temporal portion of the right retina, the nasal portion crossing over at the optic chiasm, joining with the temporal portion, and terminating at the lateral geniculate nucleus. And I had a friend that remembered this as being part of the visual portion of the thalamus as light-giving nucleus, LGN. So when we had our practical, she would remember LGN, light-giving nucleus, and so that gave rise to lateral geniculate nucleus. These fibers then connect with axons here that project directly to the primary visual cortex. And this is known as the, and it's important you remember this, do it in red, geniculonucleus cortical pathway. And really, these pathways are intuitive and kind of easy to remember if you remember that it goes from the lateral geniculate nucleus to the primary visual cortex. That's the geniculo, from the geniculus here, cortical, from the cortex here, pathway. And 90% of the visual input goes through that pathway. The other 10% of those fibers make connections with the pulvinar nucleus in the thalamus and the superior colliculus back here. So remember those, the pulvinar nucleus and the superior colliculus. And your book points out rightly that even though this 10% seems rather small, it's really not trivial. And in fact, 10% of the visual pathways comprise more fibers than are found in the entire auditory pathway. So this is, even though 10% seems rather trivial, it's not. And we'll get in, visual processing is very, very important in advanced primates, in us, and in our primate cousins. And so it's not surprising that even a small portion of the visual pathway takes a great deal more cortical real estate than many of the other sensory pathways. Now what you see here is a representation of the macaque, the monkey brain areas. And I think your book says that there's about 20 brain areas devoted to vision in the macaque. I believe that they've identified upwards of 30 areas in the cortex that are identified with visual information. So you have information coming in from the retina, going to the lateral geniculate nucleus, through the geniculo-cortical pathway to V1. So V1 is primary visual cortex. It's also known as Brodmann's Area 17, or it's called the striate cortex. And the reason it's called striate cortex is because there are axons that originate at the lateral, at cell bodies in the lateral geniculate nucleus that synapse or terminate in layer 4 of the visual cortex, and they terminate in a pattern that resembles a very thick stripe. And it's in the primary visual cortex where very, very basic information about vision arises, and that's information regarding spatial frequency, orientation, motion, direction, and speed. So remember, we talked about also the monkeys having in this MT area, this provided a sense of direction of where something is going, or a movement in a direction, that when ablated they lost that sense of movement. 3 very important names to remember with respect to the visual system are 3 pioneering researchers in the area of receptive fields. First came Stephen Kuffler at Johns Hopkins University, who demonstrated the presence of receptive fields in ganglion cells in the retina. Now, following on that, David Hubel and Torsten Wiesel went to Hopkins, where they became partners, and eventually went to Harvard. Hubel and Wiesel are famous and won the Nobel Prize for looking at visual fields in the visual cortex, and you saw some of that, or hopefully you will see some of that, in the Charlie Rose series on visual perception, and the small 30-minute clip from an MIT researcher who also mentions Hubel and Wiesel. Really important in showing in the primary cortex specific individual neurons that are sensitive to direction, that are sensitive to movement, that have specific retinal fields in certain areas and certain, the edges and shapes. Really amazing work that you should be very, very familiar with. Your book covers some of that, and again, the films that are required watching for this week cover a little bit more of that. And I will close by showing the recordings that Hubel and Wiesel did on the simple cells in the lateral nucleus that are described in your book, the on-center and off-center cells. So here they are. Here's their recordings that netted them the Nobel Prize.